Okay. Okay, cell theory. What is cell theory? Go ahead and read it for me, Cassidy.
Cell is the basic structural unit. Everything's made of a cell. It's the smallest unit of life. Your activity depends on your collection of cells. They're dictated by the subcellular structure, which are the organelles. All cells come from other cells, and continuity of life has a cellular basis.
So, out of this slide, what is the takeaway message?
Cell is the smallest unit of life.
Okay, write that down. Cell theory, the smallest unit of life. Write that down. That it will be on your exam. Most likely on your MCQ questions. So write it down, guys. Smallest unit of life, cell theory. And because why? Because it comes from another cell. So cells have certain basic components. What are they? Plasma membrane. What is the name of that tissue we call plasma membrane? That fatty acid. Phospholipid. You got a glycerol head that's hydrophilic, and you got fatty tails that is hydrophobic. They're amphipathic. That's why they're flexible, movable. Okay, so that's plasma membrane. And then we have the fluid inside of the cell. What is that called? Very good. And then what is the nucleus? What does the nucleus contain? DNA. What do we call a bacterial cell nucleus? Yes or no? No. This only applies to eukaryotic cell. But if you're studying bacteria, what do you want to call that bacteria? Where we keep all the DNA structure? We call that nucleoid. I think so. I think it's spelled that way. Nucleoids. Okay, nucleoids. Everybody get it? Nucleoid. So that's in bacteria only.
It's for what only? Back to what?
Nucleoids in the bacteria. Prokaryotic cell. Prokaryotic cells have nucleoids. They don't have nucleus. So let's go to the next one. So what do we have here?
Nucleus.
Nucleolus. Not the nucleus. Nucleolus. This whole thing is a nucleus. This is a nucleolus. Nucleolus is where the production of what?
DNA or RNA.
RNA, ribosomes. Yeah, ribosomes. So, and these little pores is where, what goes through these little pores to make copies of DNA? mRNA. mRNA. They are one of the RNA that goes in and makes the copy, and the central dogma is to make a transcription of the DNA so you can make it a proper protein. Now here, what are these guys? Mitochondria. This is the powerhouse of the cell. They produce the bulk of the what? ATP, energy currency, universal energy currency. Now, here's the question for you. This is the animal structure. In a plant, do they have mitochondria?
Yes.
Okay, very good. So, what is this bunch of little dots everywhere? Okay, then what is this structure here?
Rough endoplasmic reticulum.
And you're going to have to know what rough endoplasmic reticulum do. What do? They make protein. They synthesize protein. Then you got the smooth part here. What do? Lysosome, fat. And what are these green sections, this green fold?
Golgi apparatus.
Golgi apparatus. Golgi apparatus is UPS, FedEx of the cell. What do? They package the protein, they label the protein, and they transport the protein. So you could say Golgi apparatus is the UPS or FedEx. Okay, everybody got it crystal clear?
What's the name of it?
Cassidy, did you hear?
Yep, I'm writing as you speak.
Okay.
What? Golgi apparatus is like
Lysosomes?
Makes and synthesizes proteins.
So here, you got these little, what is these? Peroxisome. What are peroxisome?
Those are things that break down. No.
Break down what?
No.
I'm going to write this down, and you guys are going to have to remember it. Peroxisome breaks down, right here, peroxisome, breaks down,
Yep, I think.
what is that?
Peroxide.
Peroxide. Because peroxide is toxic to our system. So they break it down to, and
No.
molecular water again. And then that gets used up right away, instantaneously. So the purpose of the peroxisome is to detoxify. And one of the examples of detoxification is these peroxide. So that's what the peroxisome do. Now, let's go over some of these microtubules. Microtubules have that 9+2 structure. You guys know what microtubules are, right?
It's basically structure.
That's what the mitotic spindles to the flagellum of a bacteria, flagella of the sperm. But these have an interesting structure. They're called 9+2 because if you get a structure like this, two, four, six. And then they got two structures here. You got sets of two, like 1, 2, 3, 4, 5, 6, 7, 8, and 9. Like that, and then you got one in the middle. And there's and twos. And this is the typical diagram of what a microtubule is. Okay? So we'll go over that a little bit more later on. But generally, that's what we talk about. And a good example of microtubule. And what are these guys? These cylindrical shape, what is that?
Centrioles?
Centriole.
What did we just say?
So let's keep on going. Look at the chromatin. What is chromatin? Condensed form of the chromosome of all your genetic trait is right in the chromatin. And what is this white covering here? What is all that stuff?
The plasma membrane?
Plasma membrane. What is plasma membrane composed of?
Oh.
Phospholipids. And then you got other structures on there. But make sure you guys remember this microvilli, microfilament. These are these right there. And then finally we get to this guy here, lysosome. What is lysosome? What are lysosomes?
They break down water.
Lysosomes have enzymatic property to break down debris, break down no good defunctional protein, junk. They pick up debris and they enzymatically cleave them and put it to recycle, or they trash it. Okay. Plasma membrane. Let's read this here. Carlos, read this for me. Three bullets.
Separates intracellular fluids from extracellular fluids.
What is the name for intracellular fluid? Cytoplasm. And then you got the extracellular fluid. Okay, go ahead.
Okay. Are semi-permeable.
Why are they semi-permeable?
Because of the phospholipids on the outside of the plasma membrane. It like acts as a barrier, and then I think there's openings to let some in now.
Okay, guys, there's transport. This is the next section we're going to have to go over. There are passive transport, there are active transport, there are vesicular transport. Those are the three different transport you need to know. Passive transport, there's several different kinds. You know active transport, there's several different kinds. They got vesicular transport, that's the third kind. You're going to need to know all three. You're going to be writing an essay on all these three, so you can make sure you know. So that's why they're semi-permeable. They play a dynamic role in cellular activity. So here it is. Here's a nice fluid mosaic model. What does it mean when they say fluid mosaic model? Let's go ahead and read that for me. Kelly, read that for me.
Double bilayer of lipids with embedded dispersed proteins. Bilayer consists of phospholipids, cholesterol, and glycolipids.
Can you repeat that again?
Bilayer consists of phospholipids, cholesterol, and glycolipids. Glycolipids are lipids with bound carbohydrate. Phospholipids are arranged into two layers, lying tail to tail with polar heads. Hydrophilic equals water-loving. Exposed to inside-outside cell and nonpolar tails. hydrophobic, is water hating.
So what are the, this fluid mosaic layer of this bilayer, what is it composed of?
Phospholipids.
Cholesterol and glycolipids.
That will be on your exam, on your, essay, open-ended. Make sure you write it down, guys. Write it down, because if you don't write it down, you're gonna get it wrong. So you got all this time till next week to study this. So, phospholipid, cholesterol, and glycolipid. Phospholipids are layered in two layered line discs. They get the glycerol head, and you get the fatty chain, the hydrophilic and hydrophobic. So you got all these heads that are hydrophilic, and inside and out, and then you got these fatty chain tails here. Here, this is a nice illustration of different components in the membrane. So you got all these integral proteins, you got these channel proteins, you got these glycoproteins, you got these glycocalyx, they do communication, they do structural, keeping the dynamicity of these structure of the cell membrane. And you can see all these intermittent filaments here, or we call the cytoskeleton, on the inside and outside. And look at this cholesterol, because we said what? Phospholipid, cholesterol, and what?
Glycolipids.
Make sure you remember that. Glycocalyx. Glycocalyx is glycokali in Greek. So, and you know everything's going to be Greek for me if it's nothing Greek. So, glycoprotein is budding the cell that provides highly specific biological marker which cells recognize as one another as sugar halo. Okay, it's like for what? Communication? Yes? Yes, communication. So there'll be more on this than what? AMP2 and the endocrine system. So what do they do on these membrane proteins? What do they do? So they transport, they have enzymatic activity, they have a receptor for the transduction. So they transport enzyme and receptor. This is going to be on your exam too as well. Then we got intercellular joining. We got cell-cell communication, and we got cytoskeleton and extracellular matrix that attach and give the dynamic fluidity of these membrane structure.
Can you go back?
Yeah.
Okay, which one?
You said this will be on the exam, but I don't have enough time to.
Okay.
Transport. It was transport.
Next, the functions of it.
Oh,
So
the
membrane
functions? Yeah.
protein.
The function?
Yeah.
Transport enzyme receptor. Then on top of that, they do what? Adhesion, intracellular adhesion, cell-to-cell recognition, and attachment.
Is this PowerPoint's on D12, right?
Yeah.
Cyto-
You got another one in the D12.
Okay.
Yeah, but it's the same thing. And then plasma membrane surfaces. What do they have? They have what? Oh shoot. Different kinds of lipids because it's made of glycolipids, right? So glycol is found in outer membrane surface. Make sure you guys remember that. Why they're on the outside? Part of the communication. The glycolipids, they do part of the communication process. And then 20% of the membrane lipid is cholesterol. We know that. That's the other component. And then you get the phospholipid itself. And then membrane junction. So this one you're gonna have to make sure you get on your essay. Okay, so you got glycoprotein active adhesives, you got membrane contours the adjacent cell, and specialized junctions stick together. So we're gonna find out what they are, the nomenclature.
Wait, can you go back
Can you
one
go back?
second?
Go back.
This is the one you have to know.
Oh, okay.
I just mentioned what they do, now this is what they are.
For the
So
last,
make sure you know what these are. This will be on your test.
you said for the 3 factors that bind those
But
cells.
these are it right here. These are it.
So you said that
I-
the,
Yeah.
so you said that the 3 factors that bind the cells together will be on the essay?
Yeah, and I just said it right here. It'll be right here.
So it's not the 3 that's listed?
This three
Uh-huh.
is this 3 right there.
Got it.
Yeah. Good. Good. Okay. Let's see here. Annie, read the 3 bullets, please.
The membrane junctions. Tight junction, impermeable junction that encircles the cell and prevent passage between cells, connect epithelial cells.
So what does that mean, impermeable junction? What does that mean?
Can't pass through it.
Can't pass through it, so it's gonna be watertight, right? So what is the next one? Go ahead and read the next one.
Desmosome, anchoring junctions scattered along the sides of cells, attach cells to each other.
So what's the key word for desmosome?
Anchoring.
Anchor.
Anchoring.
Okay, what about the next one?
Gap junction, a nexus that allows chemical substances to pass between cells.
So this is the communication. Gap junction communication, look, because it allowed channel to allow chemical substance to pass through. So you got communication process. So gap junction no more than what? Communicate.
Communication.
A desmosome, the anchor, and a waterproofing, titrating, making everything tight and sealed. Okay? So here's a good illustration of the three. Now, what is this rivet-like structure here? What is that?
Desmosome. Oh, tight junction.
Tight junction. And then you got the desmosomes here, right there. You see these little, right here? That's desmosome. And then gap junction, you got these little holes here so they can communicate and the ions pass through. So, tight junction again, look at these. Rivet-like. Then you got the, what are these? Desmosomes. They do what? Anchor. Look, they anchor.
It's like
Linker proteins. Another picture, linker protein. See how they anchor? Then you got the gap junction. Look at these pores, these channels. Look at the, write this down. Right here. Channel between cells, they're called connexons. So on your MCQ question, I'm gonna list a bunch of proteins in there, and one of them is gonna be related to the gap junction, and they're called connexon. Okay? Connexon. And that's how they're able to have shuffle ions from one section to another. I know you guys hate it, but here we are. We got another test coming up. Just stick with it, guys. It's like a roller coaster ride. Connexon. Diffusion. Let's look at this. This is the important stuff. Remember I told you guys need to know passive, you guys need to know active, and you guys know vesicular. All important stuff. So let's look at this. Simple diffusion. Does passive diffusion use ATP?
No,
Why you don't use ATP?
because it's passive.
Why is it passive?
Because they just kind of go through.
They go through the gradient,
heat
right?
energy.
Guys, I want you to use this word. Look at this. Look, everybody. High concentration to low concentration. That's why you don't use energy. You're looking at the concentration gradient. The concentration gradient on one side has the propensity to move downward to the low concentration area. It's just like me getting a pizza right now, and I just put it outside in the hall, and that smell is going to come radiate into this room in low concentration. Okay? So high concentration to low concentration. That's why there's no ATP used. So diffuse directly to lipid bilayer. Diffuse through channel protein, and required what? No energy, no ATP. Now diffusion, listen to this guy. Look, key word. Diffusion of water. Look, diffusion of water, liquid, across the plasma membrane is called what?
Osmosis.
Osmosis. Put a star on that. And if there's a question that pertains to water and is moving from high concentration to low concentration, what is that?
Osmosis.
Osmosis. And it's a form of what type of transport?
Oh.
Passive transport.
What's the difference between passive and simple?
Say that again?
What is the difference between passive and simple?
Passive
don't
and
see the
simple
word
is basically the same thing.
So the same thing?
Yeah, yeah.
Or is simple a type of passive?
Yeah, yeah. It's all passive. Yeah, Kelly needs to get in. But Rockman and Yushida don't want to let him in. Just open the door. Someone's at the door? No, okay.
Got a call.
Okay. Let's see here. Diffusion. We got two different types. You got simple diffusion, and you got facilitate diffusion. Now, what is facilitate diffusion?
Meaning that it goes from
Yeah, the simple diffusion, you're going from concentration gradient, right? Facilitate diffusion, what does the word facilitate mean?
It's to like to grab or like to take some.
From the concentration gradient also. So, read this for me, Carlos.
Transport of glucose, amino acids, and ions. Transported substances are moved through the plasma membrane by binding to protein carriers or pass through protein channels. Does not use energy and move substances down in concentration gradient.
Look at that. Move substance down in concentration gradient. That substance is glucose, amino acid, or ion, sodium or potassium, or chloride, or something like that. Those are ions. So you got not only ions, but you got glucose, you got amino acid, that's the same thing. You know, like you got glucose, you like glucose and, I don't know, what glucose and potassium. So this is facilitate diffusion. So now you got two different types of classification of passive transport. What is the first one? Simple. And the second one is?
Facilitated.
Facilitated. What is the difference between the two?
No,
They both use concentration gradient.
Facilitated binds to a protein or goes through a channel.
See? Yeah. Facilitate use these.
Oh, facilitated binds to a protein or goes through protein channel.
Yeah, look, facilitated, just like the word implies, you facilitate, how do you facilitate? By using what?
Protein channel.
Protein, look, right there. Protein carrier, protein channel. That's the difference. So, let's see here. These are all different examples. You can see that on your own slides when you go over and look at different channels. You got the simple carrier mediated, you got channel mediated, you got osmosis. And what is this channel that used to fluctuate water? What is that called?
Osmosis.
Aquaporin.
What? Oh, aquaporin is the.
Aquaporin.
Somebody has.
That's the,
like,
aquaporin is the channel protein.
So osmosis, what is osmosis? Go ahead and read it for me, Sam.
Occurs when the concentration of a solvent is different on opposite sides of the membrane.
What does that mean?
So there's a barrier and then, what's that? So this is the thing that we did with the blood. I don't know, like.
One side is going to have a highly concentrated ion that's going to be hypo or hyper, and the other side is going to have normal. So where's the water going to move?
Towards.
So diffusion of the water crosses the semipermeable membrane, and then we classify this as what?
Osmolarity.
Osmolarity. Osmolarity is the total concentration. Solute. What is solute? The ions, right? Particles in a solution. You remember the solution is what is a component of two components, solvent and solute. You guys remember that? Okay. You guys fall asleep on me. What is unbound water molecule will move from the hypotonic solution? What is hypotonic? Very, very little amount of solute. Where the water molecules are abundant of hypertonic solution, what is this? More of the
More.
solute. And where they are rare. So this diffusion of water across a selected permeable membrane is a special case of passive transport called osmosis. So you can see hypertonic, remember I told you, water loves salt or they love protein or where the high concentration of the ion concentration is. So where's the water going to move to? That side where it's hypertonic. That's how our dynamic, our cells and our body fluid system work. Tonicity, we just went over tonicity, right? I just went over tonicity. Go ahead and read this for me. Lenny, read it for me, please.
Cells often find themselves in hypertonic or hypotonic environment, yet they do not shrink or swell even though osmosis is occurring. The cells counteract osmosis effects by using active transport processes to pump water in the opposite direction as needed. The only cells in the body unable to counteract osmosis are the RBC.
Red blood cells.
To protect the red blood cells, the plasma must be kept isotonic with the intracellular fluid of the red blood cells.
And you guys, during your lab exercise, you guys know the importance of why it has to be isotonic, right? You keep the rigidity and the shape of these red blood cells. Now, we're talking about passive transport. Now, if you're looking this right here, it says the only cells of the body unable to counteract osmosis is red blood cell. Why do you think that is true?
Because water contains oxygen,
Well, first of all, what does red blood cell do anyway?
transport oxygen.
Carbon dioxide, nutrient, and whatnot, right? That's the whole purpose. They run off of temperature and they run off of acidity, not the tonicity. Okay? Red blood cell run off of acidity. That's why you have your pH, and they run off of heat, but they do not run off of tonicity. Okay? That's basically what it's saying right there. So the tonicity of solution. We already went to. Do I need to explain this?
No.
Take a pic of it, whatever you got to do, so you can read it. Everybody knows, right? Clear? Crystal clear? Yes? Okay. Oh, it's already time. Okay, so isotonic. Hypertonic. What do we call this fancy word?
Crenate. Oh,
Crenation.
Yeah.
Yeah. Crenate, they shrink and they prickly, they crenation. And then we got hypotonic. What happened? The
Lyse.
fancy word, lyse. And then you got filtration system. Filtration is, what do we see this pressure? You got two different types of pressure. You said atmospheric pressure, and then we said hydrostatic pressure or fluid pressure. That's what basically what it is. It's a pressure outside the cell wall that's liquid, okay? That's hydrostatic pressure. And the pressure gradient pushes solid against containing the fluid from higher pressure to lower pressure. Now we go active transport. Why does the active transport use ATP? Why do you think this type of transport use ATP?
Because it requires.
It's going against the gradients.
Going against it, what does that mean?
It's going from low to high.
Yes, but what does that mean in a cell?
Needs more energy to be pushed across.
Yeah, it's like a simple analogy is this. Let's say this room, we have 1,000 people in here, all stuffed to the corner, and you want to put more people in here, what are you going to have to do?
Shove them in.
You're going to shove, you're going to use energy to shove more people in there. So that's what's going to happen. Mother.
Have a great day, Mr. Kennedy.
Anyways, happy Halloween.
Yeah, that's it, exactly.